All right, guys. So we're going to finish the biotech unit today. We have two short things to talk about. We just finished learning about PCR, right? And that's a very essential tool when we talk about molecular biology work. We also talked about sequencing and how we can use a PCR product, send it to a sequencing core, and then they can sequence our DNA for us and give us back a sequence. This is really important novel research as well. But there's one more thing that we can do with PCR. We can PCR certain gene fragments, like we just did, right? We just did PCR. And we can use that product to do something called genetic engineering or molecular cloning. So we're going to talk about that, and it's a really essential part of medical biotech as well as research so that we can use proteins and genes in the lab that we otherwise may not have access to. So we don't like to use animals in research, right? I used mice, and I just did not like hurting mice for research, but I had to do it because that was the only option we had. We can't really study directly on humans, so if there's a choice, we would like to either do cell culture, do culture in dishes, or we would like to use molecular devices only. So we're not really hurting anything. We're just making it recombinantly. And that's what we're going to talk about. So, yes, this is a glowing cat. This is not a fake picture. There were scientists who were trying to study a gene for a disease called AIDS. I'm sure you've heard of AIDS before. And they inserted a gene that made this cat resistant to the cat form, the feline form of AIDS. And usually, when you insert a gene or change a gene in an organism, to make sure you know that you changed that gene, they add a GFP tag to it. Remember, we talked about the GFP tag. It's a jellyfish protein that fluorescently glows. So, when this cat is highlighted with a blue wavelength light, you will be able to see the glowing of this cat and only particular tissues because that's what the gene is affecting. So we use this method of genetic engineering to not only help us in making proteins, but also in scientific advances in medical research. So making things glow is always fun, and we always like talking about it. So here's one example of a glowing thing. All right, moving on. So genetic engineering, what we call molecular cloning, is one way that we can use PCR again to help clone a very specific gene into bacteria cells or also yeast cells. But how do we do this? It's actually quite complicated. I did it in the lab about 18 different times with different proteins that I was trying to work with in the lab. It took several weeks every time I tried to do it. So let's talk about how you do it. Genetic engineering, when I say that, that means direct manipulation of genes for practical purposes. So we are manipulating the gene right then and there for some purpose. Usually, to work with specific genes, we have to make it. And we use it with well defined segments of DNA so that we can make identical copies of it. And then we clone them, basically. It's DNA cloning. Most methods of the cloning pieces of DNA in the lab share general features. The methods are literally almost exactly the same for every single one that you're doing. It depends on what plasmid you're using and what gene you're using, but the whole raw process is the same. So let's talk about plasmids, because you need a plasmid to make molecular cloning work at all. So there are bacteria that contain these little things called plasmids. Yeast also have these as well. They are small circular DNA molecules, and they're going to replicate separately from the bacterial chromosome. So we know that bacteria have spaghetti messes, right? We said that they have spaghetti chromosomal DNA. It's kind of just all over the place. That's the bacterial chromosomal DNA. They also make these plasmids, which are little circular DNAs. You can see in this picture here the plasmids. They are still DNA. They're just separate from the chromosomal DNA. And what scientists have realized is most of the time, these bacterial plasmids are mostly used for like what happens in antibiotic resistance. I'm sure some of you have already heard about that. We talked about it, that certain bacteria are becoming immune to the antibiotics we have. One of the ways is because of the ability of these plasmids to copy and reproduce, just like bacterial DNA chromosomes do when bacteria rapidly reproduce. So they are still a form of DNA, but scientists discovered that we could isolate these plasmids. Very specifically, we could get these plasmids out and we could use them to our advantage. So we have hundreds of plasmids, guys. Hundreds. You can buy them. We can order them right now. And you have specific ones based on like antibiotic resistance, based on restricted enzymes that are present. You can molecular engineer basically whatever plasmid works for your experiment. We usually bought plasmids that either did a certain thing. So maybe one plasmid had the ability to make the protein more soluble in a liquid. That's possible. It depends on what you're looking for, but there are many options for a plasmid. But to clone the pieces of DNA, we need the plasmid because we're going to insert our DNA, our gene target, our foreign DNA, they call it, into the plasmid. The resulting plasmid that now contains our gene of interest is called recombinant DNA. There are specific protocols in labs where we have to report, are you working with recombinant DNA? If so, you have to report it here. So our lab had to specifically say, hey, yeah, we're making recombinant DNA. I don't know why they were so nitpicky about it, but I think it's because it's not a natural source of DNA. We have engineered it. So they like to know those things. So it's not a bad thing to have recombinant DNA. It doesn't really hurt anything. But because it's foreign and it has foreign DNA in it, they just make sure they know it's happening. All right, so recombinant DNA technology. It's very common. Almost every lab in the world does this because at some point they want to work with their protein of interest or the gene of interest they're studying. All we're going to do is take a plasmid. See this little circle plasmid DNA here? We're going to cut part of it and we're going to insert our gene and then we're going to ligate the pieces together. You guys know about DNA ligase. We've talked about it. Remember, it's the molecular glue, the glue stick that sticks the DNA back together. And then we're going to shock our bacteria that we use. We like to use E. coli a lot. And that when we shock them, the E. coli will just absorb or take in the foreign plasmid, and then it will be in the bacteria. So here's a picture, a bacterium. The plasmids are in there. We're going to isolate that plasmid out, and we're going to copy or clone the gene of interest into that plasmid. How do we get our gene of interest? Guess what we use? PCR. The polymerase chain reaction, like we just did. We're going to have primers. They're going to stick to a very specific part of the DNA, and that's going to copy our gene of interest. And then we're going to use very specific enzymes. You all know that an enzyme is a protein, right? It has a very specific job. Most enzymes are responsible for cleaving or cutting. Not all of them, but a lot of them are meant for cutting DNA at certain specific places. So we're going to talk about those enzymes. The enzymes can cut very specifically your gene of interest at very particular points. We can also cut our recombinant DNA plasmid at very particular points and then glue them back together, just like pieces of a puzzle. So we'll then have a recombinant bacteria because we're going to insert this newly made recombinant DNA into the bacteria. The bacteria will then grow. They reproduce very quickly. Remember I told you they grow overnight in a 37-degree incubator, and they duplicate and replicate extremely fast. There are many types of research that have been using cloning things. So one, the basic research and applications for gene and pest resistance in plants, that's common. For bacteria that we can hopefully manipulate to eat oil, remember we talked about those that already do eat oil. What if we could take the gene that allows them to do that and put it in other bacteria? And now we have many species that can eat oil and clean up oil spills. There's also something called the human growth hormone that usually treats stunted growth. We can use it for that. Remember we talked about insulin creation. There's also the production of protein that dissolves blood clots in heart attack therapy. There are many ways that we can use the cloned genes and proteins to our benefit. Okay, so in gene cloning, the purpose of it, the main goal of gene cloning, is to produce multiple copies of a single gene. We want a lot of copies of a single gene. Yes, PCR does that, but we can have bacteria help us do it. The plasmid that carries the cloned DNA is called a cloning vector. So, I don't know why, but plasmid and vector is an interchangeable word, okay? So you can call something a plasmid and a vector. It's the same idea. See this circle picture on the right here? That is a particular vector. Its name is always displayed in the middle. It also tells you how big it is. So this plasmid just happens to be 3,353 base pairs. They call it Fram. Cool. Whoever names these things, they decide what it's called. You can see a bunch of little letters all the way around the outside of this circle. All those little letters are coding for very specific restriction enzymes. These enzymes are going to be where we can cut out parts of that plasmid and then insert our DNA of interest. All right, so how do we do this even more? So, restriction enzymes. Let's talk about those for a second. So we use bacterial restriction enzymes to cut DNA molecules at very specific DNA sequences. These specific sites are called restriction sites. Now, usually, a restriction enzyme can make many cuts, or it could just do one cut. And that will give us fragments. And usually those fragments you can run out via gel electrophoresis, which you guys just are going to do next time. But gel electrophoresis, remember, will separate DNA bands by size. So we can figure out if we have cut out certain parts of our DNA via gel electrophoresis. So in the picture here, look, on the bottom here, plasmid circular DNA. We're going to insert our target gene. But before we can do that, we have to cut the plasmid. The plasmid has something called an EcoRI site. That is a very specific enzyme. It happens to have one cut right where there's a G and an A. So if you look at the picture up here on the right side, it says EcoR1. We can buy this enzyme. I can buy it from NEB Labs. NEB tells you, hey, look, see where the arrow is in this sequence? This very specific sequence, GAATTC. It will only cut that DNA right between the G and the A on both sides of a double-stranded molecule of DNA. That's why these restriction enzymes are very specific. It doesn't just go cut the DNA wherever. It has to be a very specific sequence where the restriction enzyme will bind and cut it, just kind of like scissors, okay? So if I cut this EcoR1 site right between the G and the A on both sides, it allows us a spot to open up in the plasmid where we can insert our target gene. The target gene also has EcoR1 sites. So that way, we can glue it back together. And with DNA ligase, it's very easy to glue back together. So when I would do it in the lab, we could do it overnight or we could do it in an hour. Just kind of depended on what enzyme you were using and how fast you wanted to go. All right, so there's another example here listed, NDE1. That's just a different enzyme, a different restriction enzyme that you can buy. See how this one has a little arrow that's going to cut in between the CA and the TAG. It will only ever cut at this sequence of DNA in between the CA. Wherever the arrow is, that's where it will cut. Okay, so without restriction enzymes, we cannot do molecular cloning. We need the polymerases and the enzymes to be able to cut specific parts. All right. So we need to identify where the following enzyme will cut the DNA segment. So I've given you two examples here. There's BAM-HI-HF. HF stands for high fidelity. You don't need to know that. BAM-HI is a really common enzyme. Where would this cut? Okay, so think about that for a second. Look at this picture. Where would this enzyme cut this DNA specifically? You can see there's an arrow, and wherever the arrow is where it's going to cut. This is going to cut in between the two G's. So in this very particular sequence, GGATCC, BAM-HI is going to cut it right where that G is and the other G. If we look at SMA1, this is also a very common enzyme. He's going to cut right in the middle. CCC, cut. GGG. All right, so this is known as a blunt end. Remember, I think I talked about this once to you guys before. Blunt ends are a lot harder to stick together with ligase. I don't know why. There's no sticky ends here. That's what they call them. So there's no overhang. It takes a lot longer. These usually go overnight before it'll stick back together. You'd rather have a sticky end, which is what the BAM-HI is. All right. So after this, you guys are going to have to work on some homework assignment. It's a Google assignment where you're going to have some problems that you're going to have to figure out where restriction enzymes go. So pretend that a restriction enzyme is like a pair of scissors, and you're going to cut the DNA, the circular DNA, wherever the enzyme falls. So this is number 1 of the homework you're about to do. I just want to kind of point out stuff to you before you look at it. You're like, I have no idea what I'm doing. So below is a restriction map of a very particular plasmid. They call it PGen101. The total length of this plasmid, that means the whole circle, is 20 kilobases. So using this map as a guide, what we want you to do is determine the size of fragments if you were to cut with EcoR1, for example. So if I were to cut at EcoR1, which happens to be where EcoR1 says here on the bottom left, there's only one EcoR1 on this whole map, right? Okay, so if I cut right there, what would be the whole size of that fragment if I ran it in a gel? It should be 4 kilobases plus 2 kilobases plus 6 kilobases plus 8 kilobases. Guess what the total length would be? 20 kilobases for EcoR1. For BAM-HI, sizes of the fragments obtained. So there's many BAM-HIs here, right? There's 3 different places where it's going to get cut. The enzyme's going to cut it here on the left side. It's going to cut it here above that side. And one more time next door. It's kind of like a quarter turn of a circle. So if I were to add all these fragments up, let's see. I would definitely have a fragment that's 2 kilobases. I would have a fragment that's 6 kilobases. And then I would have a fragment that kind of finishes the half circle with 8 plus 4. So I would have a total of 3 fragments this time. 3 cuts, 3 fragments. So you would just have to tell me the sizes of each fragment and how many fragments there are. Now, if I were to use EcoR1 and BAM-HI, how many sizes of fragments would I get? So EcoR1, that's just going to cut it one time. The fragment size we would get would be 20 kilobases. However, we have BAM-HI in here as well, which is going to cut that giant fragment that was just made with EcoR1 into 3 spots again. So you're going to end up with some similar fragments. You're going to end up with a 2 kilobase, a 6 kilobase, an 8 kilobase, and a 4 kilobase. Every single spot here where there's an enzyme is going to be chopped up. So just look at the spots in between. From BAM-HI to EcoR1, there's an 8 kilobase section. That's 1. From EcoR1 to BAM-HI, on the left, that's 4 kilobase. In between the BAM-HIs, there's 2 kilobases. From BAM to BAM on the top, 6 kilobases. So every time you see a segment here, we just cut it all. So you're going to have 4 segments, and each one correlates to a size. I know it's hard to see that because I'm not physically there in person and drawing on the board. However, hopefully this helps you figure out kind of what the questions are asking you. There are 2 other examples like this. Just look at the map, pretend you have scissors and you're cutting on the line. Then tell me how many fragments you would get. This is what we do in the lab. We determine, okay, so what enzyme should I cut with? I need to figure that out because I want to know where I want to put my gene of interest. So this is always the first step. Okay, so this is kind of a picture of what you're doing. Here's your plasmid up on the left side. Hang on, let me see if I can grab a pen here. Yes, here's a plasmid. We want to cut this plasmid right where this little line is here. Okay, so we're gonna digest it with a restriction enzyme. You can do that in a PCR machine too. Enzymes are active at 37 degrees, like our bodies are. So as long as the PCR machine is set to 37 degrees, he will cut with an enzyme. He'll cut your DNA. So we cut it. Here it is. We cut the plasmid here. We also cut the plasmid here. So now there's kind of like a hole. We can then take our target gene. Let's say, for example, it's the gene for insulin. We cut out the whole gene for insulin. We PCR it, get that product, and we pop it in to our plasmid and join together with a ligase, and voila, look, a completed plasmid. As long as the plasmid is ligated back together, it will form a circular DNA plasmid that can be put back into a bacteria cell. Yeah, get rid of my pen. Sorry, guys. Okay. So here's the big picture. Bacterial plasmid. It's circular. There are restriction enzyme sites in this plasmid. We can use enzymes to cut at these specific restriction enzyme sites, which then splits it in half. This here is called a sticky end because there's a little bit of an overhang. We can then put our fragment of interest, our gene of interest, and pop it in to where we just opened up that plasmid, and now we have a recombinant DNA molecule. It's kind of like a puzzle. You're putting pieces of a puzzle together, and in the end you end up with a recombinant plasmid. What do we do with that recombinant plasmid? Okay, so we have to get this plasmid into a bacteria. We're going to say E. coli. E. coli like to grow in some form of nutrient broth. We then grow the E. coli, and we shock the E. coli with this plasmid, this DNA. And the DNA will get absorbed by the bacteria, and the bacteria will then start making copies of themselves and copy that DNA amongst themselves. You then have to determine which bacteria took your plasmid, because they don't all do it, sadly. Only some of the bacteria can uptake that plasmid. So we take those cultures, we call them, and grow them on an LB plate. An LB plate is like an agar plate. And once you put the bacteria on there, if there is a bacteria that contains the plasmid, it will grow on this plate because I put something called antibiotics in there. You all know what antibiotics are. And most plasmids contain an antibiotic-resistant gene. That way, if I grow this on an ampicillin plate, ampicillin is pretty common. If I grow it on an ampicillin plate, that means only the bacteria that took up my plasmid are going to be able to survive ampicillin. And therefore, they will grow on the plate. So you see these little colonies. They call them colonies. Each colony is going to be a bacteria. Any bacteria that did not uptake the plasmid, they will die because they are not resistant to ampicillin because they did not take up the plasmid. So as soon as I know that I have colonies, I can be hopeful, hey look, my bacteria that I just shocked with this plasmid took it up, and there they have it. As long as that bacteria has the plasmid, you are successful. You have cloned successfully. And then you can start making your protein in the lab. I know this seems like a lot, it seems complicated, but once you see the short video at the end of this, I think it'll make more sense. You just need to be able to know that recombinant plasmids can be made in the lab so that you can make protein instead of using animals or organisms to study your protein instead. Okay, so once your colony, you found it, you put it into a culture, which is a big, I guess it's a big culture tube, but it's in a flask where it's got all the nutrients it needs to be happy. And bacteria will grow like crazy. The bacteria, you then induce them. And when you induce them, they'll start making your protein. Now, these bacteria don't just randomly start making the protein. You got to tell them to by inducing them with a chemical. The chemical will trigger the bacteria to say, make it. And then they start making it. And then the cell will have tons of your protein because it's going to get transcribed into mRNA, just like we learned. mRNA that gets translated into the protein. And the cell now has loads of proteins that you can isolate from the cells. And voila, you've just made a factory for making a ton of the protein of your choice. Okay, so I'm sure you've seen these before. The GFP protein. These are some examples of gene cloning that has worked well in animals. We cannot do it in humans. I'm sorry, it's not possible. Remember, we can't study on humans, but we have done it in mice. We've done it in frogs. You saw we did it in cats. We've done it in salamanders. Normally, the gene of interest, it glows so that you know that you've altered the gene. So it's kind of like a marker. And you can mark cells too. Like if you're doing cell culture, you can mark cells so that they glow so you know they took your gene up. A lot of the things that are glowing here on the screen are because they were messing with proteins or expression of genes in muscle, certain muscle layers or muscle tissues. So they just wanted to see, especially with development, how did those muscles move around? It's just like a marker, a molecular marker. Okay. So that's it on cloning, guys. Let's see if I can get through CRISPR real fast. So molecular cloning is a little more complicated. You're going to watch a video after this video is over, a little bit easier to understand what cloning means. So that'll be a 2-minute video. It's very short. But when you're done with that video as well, you are going to summarize the steps that you need to do for molecular cloning. It's like the big picture steps, guys, not the individual steps. Like, what are you trying to do in molecular cloning? We're trying to take a plasmid from a bacteria, and we're going to try to put a gene of interest into that by using restriction enzymes and DNA ligase and PCR product. And that's it. And then you try to transform it into a bacteria so that you can then make it at your will. So just try to summarize the main big picture, okay? So let's try to finish up CRISPR real quick. CRISPR, we've talked about it before. CRISPR-Cas9. It was actually adapted from naturally occurring genome editing system that happens in bacteria. Bacteria use CRISPR-Cas9 as kind of like an immune defense. CRISPR, remember, is like molecular scissors. You can cut out whatever you want and put something in if you wanted. But for the most part, CRISPR-Cas9 is simply just the scissors. Cut out the bad. Okay, cut out what you don't want. Bacteria were fancy and they could do that. They could cut out bad viruses that happen to be in their genome. And by doing that, allowed them to create kind of like an immune defense for themselves. Somebody discovered this and decided to see if they could use it. What CRISPR stands for, it's a really long name. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Yeah, that's why we call it CRISPR instead. There's a very specific protein associated with this CRISPR called Cas9. It's associated protein 9. There's a small piece of RNA that we need. It's called a guide sequence, and you need this guide sequence so that you know where the RNA is going to bind so that it only cuts very specifically right where you tell it to. That way it's not going around and just cutting all your DNA. So this won the Nobel Prize in Chemistry fairly recently, in 2020. In fact, by 2 women, Jennifer A. Doudna and the French scientist Emmanuel Carpentier. Those 2 are responsible for the discovery of CRISPR, and they call it the greatest weapon in genetic technology because CRISPR is going to change the world, guys, and I mean that. It is gene scissors. We can cut out whatever we want and replace good genes. That's why we talked about it when we talked about genetic disorders. If we could cut out the bad gene that's mutated and replace it with the correct one, that is absolutely going to change how we see medicine. All right, so CRISPR-Cas9. When you introduce it into cells, you simply inject it or you put it in a solution and let the cell uptake it. There is a very specific guide RNA you need. You can make them, so they're very specific. And it will follow where you tell it to go. The guide RNA says, hey, go to this very specific section of DNA. And when it goes there, the CRISPR is going to go find that guide RNA. And when it finds it, the Cas9 complex that's attached to the CRISPR is going to cut it right at the targeted location. Once you cut the DNA, we've talked about DNA repair. Remember we said that DNA has ways to repair itself via polymerases or exonucleases. Those guys are going to jump in and repair the DNA that's broken because we broke it on purpose. We cut it, right? As long as the DNA that you need to repair it with is around, it will repair it. If it's not around, sometimes it will just seal up But most times, as long as you insert the correct gene sequence in there and it's floating around in the cell, it can just add the correct sequence. So that's why CRISPR is revolutionary. So here's a picture. We make a guide RNA, that's this purple thing, and it's going to go stick to a very specific sequence. Kind of like primers, guys. Like, remember we talked about primers and how they stick to a very specific sequence? That's kind of what the guide RNAs are. As long as it's stuck to the correct complementary side, we can have Cas9, this big blue bubble thing. Kind of looks like a polymerase, right? He can come in and he is going to allow the cutting on both sides of the DNA. See how this is opened up here? And on two sides, there's scissors? Yeah. We can cut it on both sides, not just one side of the DNA. Once that's done, if there's a mutation, for example, there's a gene disrupted by mutation. If you look on the right, we can cut out that bad mutation, add a donor template, and insert the DNA, which is then repaired through normal DNA repair processes. So we've just fixed a mutation using this system. Okay, so how can we use this method? 1, we can modify immune cells as a form of cancer treatment. We can cure sickle cell, which remember is a single point mutation. We can fix hereditary forms of blindness, HIV and AIDS, cystic fibrosis, muscular dystrophy, Huntington's, COVID-19, and there are literally many others that we could use this for. So that's it on CRISPR, guys. It's a lot more complicated than that, but I just wanted to give you a brief overview of like what it does and kind of what it will do in the future. Okay, and there's a short video we will watch next class when I am there. You're not going to watch this today. And we'll talk about the other part later. So that's it on the notes today, guys. Sorry it was so long. If you don't get through all of it, don't worry. So thanks for listening, and I will see you next time. Bye.